causing that depolarization, which incidentally, dragging behind it as it opens the channel. There's actually a site inside that this outer plug piece that's being described here comes up and blocks the channel. Okay? Essentially pulling a bucket behind it and blocking that. So there's your description of your voltage gauge sodium channel. Yeah, can you go back to one? You're like, wow, he put other stuff in there. Yeah, I know. Because I'm okay. We also talked about the nicotine acetylcholine receptor, and in doing that one, right, we found the acetylcholine, which had, what's the charge on the nitrogen? On the acetylcholine head group? It's positive, so that actually helps attract in this instance, and then the carbonyl on the ester bond in the acetylcholine, that actually fits in right here, attracted to the hydroxyl and the serine, and this forces opening of this structure, and the pore then in its open form allows for the entry of the calcium, the sodium, a little bit of calcium, minor bit of chloride, of sodium, potassium, sorry. There is an agonist, right, blocking it, an antagonist, leading to paralysis and death. So we move beyond that. Integrins interacting with an extracellular domain of an arginine, glycine, aspartate containing type proteins. So there's a specific amino acid sequence that these integrins like to react with. As they react, again, I mentioned that there's a specific diameter. There is an alpha and beta on these, and there is a specific side that interacts with one versus the other. The alpha has the calcium binding sites that you're familiar with when you're talking about muscle contraction. This is also describing what happens when I interact in a way that I can now start doing cellular division and adhesion functions. This could be on the test.
Good to know.
It's good to know some things, right? But what could be there? Well, there's a lot of stuff in here. Let's see if we can jump to a few useful facts. When we started this, we talked about that hormones, when they're hydrophobic, they have the ability to largely descend right through the membrane, passive diffusion, just goes all the way in. They actually go into the nucleus, where they actually go to hormone binding receptors. And those hormone binding receptors, once bound, produce a hormone response element that allows these to bind to the DNA. That's going to ultimately alter transcription. Okay. Sometimes they're promoters, sometimes they're inhibitors, but that affects then the translation of what's going on. In this instance, we're creating new transcription of mRNA, and hence we get new proteins that are in that altered function. Okay. What are some things that might be hydrophobic enough to penetrate all the way through? What type of molecules might you guess would have the ability to do that?
Steroid.
Non-polar sterols are typically the ones we see there. There are other sterol-like ones, meaning retinol is also got a site right down there in the nuclear site affecting transcription. So good ones to sort of be aware of. But there is a receptor. The receptor is a protein just like the GPCR was a protein, just like the tyrosine kinase was a protein. But this one's now inside of the nucleus. Okay? It's a little bit different play there. I put this image on here just so that you could see something about what I meant and was trying to explain to you. The flagella inside of a bacteria, how does it know where to get food? There's some attractant it's going through it actually on a rotation where it does a run, it'll move forward, and then it has a rotation where it goes into just the tumble position, and it actually randomly moves around, and it hones in on the attractant. How is it doing so? It actually is promoting this phosphorylation event, which ultimately leads to downstream this spartate being phosphorylated, which affects the motion of this rotary element. Okay, this is an electron transport chain that's powering it with protons. There is an ATPase that sits right there. This is exactly the ancient form of what we would say is the ATP synthase. It's just a little bit different in its design. So, very similar though. Modern one still has the C ring, we still have, there's a lot of pieces that are all the same.
To confirm, is that like a tyrosine kinase cascade, or is it more simple than that?
It's more simple than that.
Okay.
Yep. Fairly straightforward. It doesn't want to affect anything but the motion of the flagella, so it's not got a lot of pieces in between point A and B.
Cool.
Okay. And there's only a few pieces on the flagella. Okay. Other similar signaling molecules. I always think it's interesting, jasminate. Jasmine's actually considered to be a plant hormone. Every human that I analyzed their blood, I found jasmine. Every one. I don't know where we get so much exposure to jasmine. I'm not talking jasmine rice, I'm not, just there. It's like, really? I never could figure out why. One day when you tell me, that'll be great. Yeah. Prostaglandins, what were prostaglandins about?
Inflammation.
Inflammation, how does he know that? What was the precursor to it?
Arachidonic
An
acid.
icosanoid, arachidonic acid, inflammation response, okay, causing pain, fever. Okay, good. What was the rest of them? Tell somebody else you know what the rest of that icosanoid response was.
So I'm gonna say that's correct. I can never think of all of them. I always remember prostaglandins because I had to learn that one for immunology. But I had troubles with the other ones.
Thromboxane is for blood clotting.
Thromboxane is blood clotting, and then leukotrienes is smooth muscle contraction.
Prostaglandin has a 5
What was that?
and
Yes. Leukotrienes is the smooth muscle contraction helps with like vasodilation, basically getting more blood in. There's the signal molecules that are in immunology or please no.
Plant ripening,
So those are
seed germination, all driven by signals. You don't need to know everything there, but these are all signals that are part of a perception of how does the world outside, how are we communicating that information onto downstream targets. This is the last highlight section we want to go through, and I will quickly hit a couple other things about cancers behind that too. But this one right here, I altered this slide because I went and double checked to make sure I got it right. This is, the textbook image was wrong that I had from it. This right here is vasopressin. Vasopressin was the one that we just looked a little bit about, right? Vasopressin is going to do what to your blood pressure? Increase it. Okay. So, yep. So it's able to recruit aquaporins. What was an aquaporin's role?
To transfer water.
Transferring water. Yeah. So turns out that it does that by activating the pKa. This looks very similar to the epinephrine or beta-adrenergic receptor. Well, these look identical then, so how are they different? Location, 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 right? Light. What is the GPCR that interacts with light? Rhodopsin, okay? And yes, that means that the guy I went to graduate school with and worked with for 7 years, that I'm good friends with, that he solved that structure that we make drugs off of. Yeah, that's what made him famous. Yeah, that's what we're talking about. Rhodopsin, we're gonna take a look at this really quickly, but it's interacting instead of on a, what does AC stand for? Adenyl cyclase. It's actually acting on a phosphodiesterase, which then is making the cyclic GMP go down, because I'm cutting it up, which then actually that molecule, which was here, is then affected, and I end up closing a
Sodium channel.
a channel. Yep. Now, there are other odorants. One here is acting on a, what is PLC? Phospholipase C. Okay, remember that was cutting off, and then we were finding things like inositol triphosphate, right? We were talking also about adenyl cyclase. Okay, so IP3, CAMP, we're familiar with those. Opening channels in this case, instead of closing them. Here we're talking about sweet taste. By the way, there are ones that are not GPCRs that also function in your taste buds. Here we're talking about the sweet taste, and it happens to affect adenyl cyclase, raising CAMP, causing pKa to go up, which actually phosphorylates a channel, and that leads to closing that channel, okay? So all of these are downstream targets. This is the summary one I'd have you know. Light, rods, cones, beautiful. You can see the GPCR. I'm not gonna go through the clip, but yes, light's coming in, it's hitting the proteins inside of these structures. We are looking at the rods and cones, which are rich inside of rhodopsin. Cis11, going from cis to trans, which is ultimately a very large motion on that fatty acid. It's inside of the GPCR that's actually then changing and saying, okay, I'm going to change shape as a result of that signaling onto the G protein, kick off my GDP, right? And now I have GTP bound alpha subunit, which is now dissociated, affecting that phosphodiesterase. We just talked that through, right? Closing and shutting down the cyclic GMP causes it to fall off, which actually ultimately closes the channel. No more of the sodium, calcium running through, and that causes a depolarization of it. So that's a weird phenomenon. What I just told you is that you're constantly pumping in your eyes what? Sodium and potassium around so that you can maintain your sight. Kevin, you had a question?
Yeah, I just wanted to make sure I understood. The rhodopsin versus retinol, like, because vitamin A, right, is like the retinol in there. Is the rhodopsin itself is what gets straightened down the cis to trans, or is it retinol?
So rhodopsin is the GPCR. It's got 7 transmembranes. Inside of rhodopsin is
Where the retinol gets
where the retinol
okay,
is
so the
moving
actual
from its cis11 position to trans.
Perfect, okay. I got it straight. Thank you.
Yeah.
You said sodium and potassium being concentrated. What is it?
No, yeah, calcium and sodium, sorry. Thank you for clarifying. It's just my mind running. We say sodium and potassium way too commonly, right? Okay, so that makes some sense to what's going on. If I am going to reopen it, I'm going to need to, again, turn on my, what are we doing here? This is not an AC, this is a GC, guanylyl cyclase, to make my cyclic GMP, and then ultimately that'll open it. What is this right here? This RK, and then it's got this.
Rhodopsin.
Rhodopsin kinase?
No. It's
Rhodopsin.
It's the arrestor, I guess is what it's called.
Okay, so we're on that right track. So this is a rhodopsin kinase, and it's specifically phosphorylating these serines and tyrosines. And what is its goal from that? To recruit this, we could have said R. We'd be pirates then, right? But what is this doing right here? That's the arrestin.
Okay.
Wow. that was desensitization to light, right? That's how it works. Is that just the same as what you looked at for beta-adrenergic receptor? Yeah, same game, okay? And so we work on that. So do you need to know everything? No. What do you need to know? This slide. What could I be thinking about in this slide? Oh, well, let's see. What are the common things I'm working with? Am I working with cyclic AMP, adenocyclases? Got it. Okay, pKa, oh, phosphodiesterases, ion channels, are they going open or closed? Anything beyond that, you're way beyond the mark of what I need, okay? All right. Yeah, you, obviously when you see different ones of your rods and cones, those wavelengths are activated, then we assemble them just like your print cartridge to see sight. Whoa! About print cartridge with great science. No, it's been around for a while. Okay. Here's the one we talked about before, detailed one of the odorant coming in there. Some of you should probably take a shower more often. No. Great. Taste response, right? There are other ones, sour, salty, ion channels mediated, sweet, umami, bitter, are GPCRs though, okay? Cell cycle, why would that be a big deal for us?
I mean, we would need to tell, how do we tell each other that it's time to happen?
Yeah.
We discussed how like when insulin is on that pathway, when insulin binds it, it ultimately activates a kinase that tells the cells, the nucleus that we need to reproduce.
Right, right. And if I am talking about a cancer, what do we have a problem with?
Replication.
Replication. If I am talking about getting something to grow in a lab, a bacterial pathogen or something, I'm talking about replication cycles. So we study replication cycles at nauseum. And so we have different phases. The S phase, maybe you're aware of, that's where we synthesize the new DNA strands. If I'm in S phase. If I'm in G2 phase, following the S phase, what am I doing? I'm now going to copy enough copies of the proteins, right? So that I can now go ahead and do the cellular division. Mitosis is what you've always known it to be. I'm going to now pull this apart, right? Once I go there, I have the option of going out to the G0 phase. That G0 phase can actually be stagnated for a long time, potentially a lifetime of the cell, but there is a specific ability to then actually send a signal and re-enter the phase where it goes from the G1 phase, and then we start working on growth phase again, okay? What would be the advantage if you were actually working on a hormone or some insulin and you said, hey, I'm gonna have to sacrifice all these cells because I can't get it to kick my molecule of interest that I just put a plasmid in or I just put something in, and I want to keep everything of those cells. What would be the advantage of having knowledge of the ability to change these steps?
Are you trying to say that you can trick the cells to go into either the G0 phase if you want them to, or the replication phases if you want to?
I can. That's a really clever thing, right? Because if I can get everybody synced up to producing a whole bunch of protein for me, instead of investing all their energy in a growth phase where they're just dividing cell after cell, what I want to do is have them produce a whole bunch of protein product, and then I'm going to butcher them. Yeah, that's a great strategy, and you would want to understand that if you were trying to get into these different routes, right? So, cool things to start to think about. Cycle-independent kinase, okay, accumulate this as the cyclin concentration increases, phosphorylate it, then affect the threonine, and then I have a phosphatase that can dephosphorylate this, but the cyclin-dependent kinase itself phosphorylates the downstream DR, I don't even know what it does, and that goes downstream. But we are ultimately trying to affect this cycle, okay, cell cycling. And so this is a matter of phosphorylation and then taking it off. So these cyclin-dependent kinases are a big part of that downstream growth factor. Why do we see that? Again, cancer, cancer, cancer. So this is looking inside of colon cancer, right? A polyp is being created inside of your intestines, okay? So normal epithelium, small, grow, right? Large and bigger, and we call it then this cancer. What is this? A cyclooxygenase. What were cyclooxygenases affected by? I know, it's like really obvious, right?
NSAIDs.
They are. They're affected by NSAIDs, and they're affected by your omega-3s. So, that's interesting. So potentially, cancer growth could be affected by
Diet.
diet. Shocker. Now, some people swear by this. I don't go there, and the reason I don't go there, just to be transparent, I don't want the message to be confused. This is about prevention of a cancer, if you're talking about omega-3s here, and proper diet, not curing cancers. Okay, I don't think you're gonna cure the cancer because you start taking the omega-3s like crazy, okay? But those are the things we're looking at there for the growth factors that we're trying to regulate in the
Really bad.
cyclin-dependent kinase.
Does say omega-3s, are they like factors of, say, slowing down cancer from spreading? Is that kind of what a deduction would be, or the actual prevention of it starting?
So you are affecting the molecules that ultimately lead to downstream.
Like increased reproduction.
Well, that's right, because when I have damaged tissue, okay, one of the things that I do is I start to do cellular division. And so anytime I trigger an inflammation response, that's what's going on, is you're convincing the body I should make new tissue here, okay? So there's a debris removal and a tissue regeneration. And as long as that's kept in the right status, we're good. But I need to turn off,
That response.
yeah,
Yeah.
and
So
that's
that the
part
cancer
of that
doesn't grow.
omega-3.
Okay.
Okay, does that make
Yeah,
sense
that
to you
does.
now? Why that would be there? Okay. Well, that was chapter 12. We really need to move on. We're at half past, and we've got to talk all about this awesome topic of now dealing with. Let's see. So let me remind you, on the upcoming test, will there be ones dealing with these questions?
Yes.
Yeah, so we've already gone through that, okay, so know that for the upcoming exam. What else might we see? One's dealing with caffeine, okay? What questions are we talking about? Be able to draw the caffeine molecule? Kicks and giggles, what do you know about the test?
Multiple choice.
So you should probably at least recognize it. Okay,
Nice.
got it, okay. The Wikipedia on caffeine is an I learned. There's also one on caffeine stimulating memory consolidation. What is the receptor that caffeine interacts with, and what does it do when it interacts? What's the secondary messenger that's being interrupted? Be able to describe caffeine's general effects on the body and how these responses align with other responses. So you already see the xanthine class of molecules, of which caffeine is one. Notice how close it looks to adenosine, and you're like, that's not really all that close. Interesting, because it turns out that we are talking about an adenosine receptor. So, looking at what goes on here, caffeine is consumed, it antagonizes the adenosine receptor. In other words, what's it doing?
Blocking.
It's blocking its normal natural response. So when adenosine levels get high enough, you actually get drowsy, You drive off the road, you fall asleep on your book, drooling, right? Or you just go to natural sleep, which is what you really wanna do, right? So that's your normal response. Caffeine's preventing that, okay? There's your normal molecules, there's a whole bunch here, and ultimately, we're talking about affecting also GABA, we're gonna affect serotonin, so there's a lot of things here. Our question is affecting memory, okay? And so, can we affect our memory if we're affecting these questions, okay? So, I show you that not because you need to know anything there, but because we're gonna look at some of these downstream molecules, serotonin, GABA, epinephrine, norepinephrine, dopamine, okay? Caffeine has some listed pros. You're gonna be alert. Potentially reduce a little bit of pain. Increase your appetite. Oh, fantastic. Appetite control, rather, sorry. Reduce your inflammation, increase your force of your muscle contractions. Cool. Jump higher, get harder, great.
That's why people have
Rapid
I guess.
rapid respiration, which is really good for a low birth weight baby, because I can increase the likelihood that they're gonna get enough oxygen. Cons, can increase the heart rate, can make people agitated, easily angry, right? Can interrupt the sleep-wake cycle, insomnia, inhibition of the TNF alpha, which is tumor necrosis factor. Alpha is the one that destroys your cells, specifically targeting cancer cells, yeah. And it also reduces your overall innate immune system. You're more likely to get sick. There's the adenosine receptor, cool. Caffeine and antagonist to that receptor. Wow. Okay. There's another factor that we're going to look at with caffeine right here. What do you see caffeine affecting here? Cyclic
AMP.
AMP. The phosphodiesterase is being affected by caffeine. It's a blocker of the phosphodiesterase. Now, wait a second. Is caffeine specific to a tissue?
No.
Answer is no. So that means I'm gonna block all my phospholipids. That's right, all my phosphodiesterases. That's frightening, because how many pathways did you see with CAM?
A lot. You said
A
there's
lot.
2,000 downstream products, right?
That was with omega-3s.
Omega-3s,
Yeah,
okay.
but there's a lot of pathways being affected with cancer. The paper is about this topic. So lest you miss everything, this is what the paper's talking about. Let's see if we can understand it in a nutshell from this diagram. And you still should have read it. In the study, when did they give the caffeine? Before or after they gave them a chance to study? After. Gave it after. Huh. So, and they did a placebo effect too. And so then they do in the study, they show them something, and then they turn around, and after a period of 24 hours, they come in and they look at different dose levels. This is called lower discrimination, the ability to distinguish in these on your multiple choice recognition test, right? Do you recognize something as being wrong? The structure has an extra methyl group that shouldn't be there. The structure's not quite right. There's an amine there that shouldn't be there. The apple, is it right?
No.
You saw an apple, but this isn't the same apple. That was there, that's new. This lower discrimination, that's what we're talking about in this paper. Does caffeine though, and what did they see? Does it affect it?
Only in the short term.
Yeah, it does on short term. So let me show you a couple other things on that. Is caffeine a cognitive enhancer? Answers to the questions, right? Effects of caffeine on cognition reviewed based on large body of literature. Caffeine does not usually affect performance in learning and memory tasks. Wait, what did you just learn about? A side paper, and you're talking about lore discrimination. Okay. Although caffeine may occasionally have facilitatory effects or inhibitory effects on memory and learning. Meaning it could help or it could hinder it. Caffeine facilitates learning in tasks which information is presented passively, tasks which material is learned intentionally. You know you're trying to learn it, that helps. You're sitting there like a paperweight in class, that helps. Well, that seems obvious if you're awake versus you're asleep drooling on the books, right? Okay, that makes some sense. But caffeine does not work on facets where you're trying to then recall information from your working memory, your long-term memory, and access it. Why? What has caffeine done to your blood rate?
Increased it.
Increased it. Ultimately, you're alert, but you're not processing old information correctly. Your CNS is actually not functioning correctly.
Huh.
Wait, so if I'm trying to remember something, I may remember it incorrectly or not at all. That's what it's saying. Oh, so I might not do a good job on math. True. I might not do a good job in learning chemistry. True. Because if you need to access past information in order to process it, great. But if I'm just trying to learn something brand new for the moment, hey, great. Probably not an issue. Did you just learn something about it? Yeah. There's the paper you read, and in a nutshell, I want you to pay attention to, this is telling you all the other things I told you about it. My knee-jerk feeling is anything as a drug that I know is not only affecting many pathways, but also is addictive, and we can all admit that caffeine's addictive. Can you take caffeine and still go to the temple? Chuck, you can do it. Can you drink decaffeinated coffee and go to the temple? Nope. Wow, then it wasn't caffeine, was it? It wasn't caffeine, but is it still an addictive drug?
Yeah.
Be careful about endorsing in your life anything that's an addictive drug. It can always go too far. End of story, right? And if you just saw it affects your immune system, caution, right? I think the reason this paper works, by the way, is because of what we describe as sort of that aphrodisiac effect. Your brain just barely had a stimulus that made your heart race, made everything get excited for just a few moments as you took caffeine, which if that's not a regular occurrence in your life, and now you take caffeine, what is your brain trying to do? Figure out what caused that. So they do it post the study, and what's it doing? It's saying, whoa, what just happened? It's replaying things of recent event in your mind, trying to figure it out. I think that's where it has a lot to do with it. Well, good luck on your paper. I'll put those questions up again that we're trying to answer. You guys got this, okay? I'll be around to answer any questions I can.
Caffeine. I think the thing to remember is that there's 4 nitrogens,
Make
2
sure
carbonyl
you know
groups,
these answers for the upcoming test, right?
all but one of the
I just sent it
nitrogens
in the
have
chat.
Right, so that's how it's different
Oh,
from
really?
from sanitation.
Oh, man.
Most of the nitrogens are methylated. All of the nitrogens right are part of a double bond.
Chugging my caffeine over here.
Honestly, my first thought was, you know, you start with adenosine, you know, you have your